Okay, let's start with a few announcements. So, week from today, back at it again, exam 4. So homework will be due from 15 innate immunity, 16 adaptive immunity. We're going to do just a little bit of 17 today. Pay attention to what I talk about in class. There's not as much like in the book that reflects what we're going to talk about in class. And I think we'll get through the Staphylococcus pretty easy. We'll probably start that today. It's a pretty short section. That's in chapter 19. The chapter 19 homework, I'm going to push off until the final though, because I have it like all the staph and the strep together in 19, and we won't get to the strep, I don't think, for this exam. So a couple of you have already done it. Kudos if you did that already, but it won't be due until the final for that. I have to teach another lab section Wednesday morning, so I won't be available. It's not a popular time for you all to come see me anyway. I am available after lecture today and Wednesday, if that's a good time. But you know, if you've been wanting to pop in, I'll be there for an hour maybe after lecture, and I'll be around Friday morning as well, in addition this week. So that is what's to come. I know last lecture was a lot. It seemed like a lot to me, and when we talk about adaptive immunity and the antigen presentation and how B cells and T cells get activated. Remember, I really think, especially for the adaptive immunity stuff that's new to you, I think your book is a good resource. There's still a week out, so if you know, read the pages in your book that talk about, you know, MHC1, MHC2. You got to kind of just be exposed to that a few times to start to, at least I did, to start to understand it. So use your book, do a little reading. If you haven't done that before, this is a good time to start. I wouldn't say read your textbook the night before the exam, right? Night before, you're going to be concentrating on the notes and what we talked about in class. But again, if you do a little reading, it'll help you a little bit, you know, more fully understand, maybe help you remember it a little bit better. The one thing that's in your book that we didn't talk about is a section called clonal deletion. And I just was looking for ways to kind of cut down on the amount of time we spent on immunology. And that's just where it talks about how our B cell and T cells learn not to attack our self antigens. Okay, so that just talks about how B cells with B cell receptors and T cells with T cell receptors, again, that recognize self antigen, end up dying of programmed cell death, apoptosis. So that's really the only thing that we've kind of skipped over for the most part that's in your book. And again, read about that if you're interested, but we skipped it. So at the end of chapter 16, it talks about how we acquire immunity. And this is just really a little bit of terminology that you hear. And, you know, and they put two different terms together and kind of switch those terms around. Sometimes it's a little bit confusing, but these are the 4 ways that we describe that we can acquire immunity to some infection. So we either say we acquire it naturally or artificially. So what's the difference? I always ask myself, is this a way I could have acquired immunity 500 years ago? And if the answer is yes, then we'll call it natural. If it's artificial, then it's going to involve maybe, you know, some kind of technology like a syringe, for example. And then is it active immunity or passive immunity? If it's active, that means that we have the ability to make antibodies ourselves. If it's passive immunity, we're just getting those antibodies to target that infection from someone else. Okay, we're not making our own, but we just get those antibodies some other way. Okay, so if we put those together, if we have naturally acquired active immunity, that describes when we get an infection, just, we're going about our daily business, we acquire some infection. For example, I got chickenpox when I was 8 years old. And now I have a naturally acquired active immunity to chickenpox. I have memory cells, and if I'm exposed to it again, I quickly make antibodies that I shouldn't even, know that I was ever even exposed to it. Okay, so I've got good memory for that. Natural passive immunity, that's when you get antibodies, so preformed protection from someone else. And so when you get those antibodies, right, they work immediately. The problem is you're not making them yourself, right? You get them from someone else, they last a little while, and they may degrade. The really only example of this where that happens naturally is by breastfeeding. So a breastfed infant will naturally acquire antibodies. What kind of antibodies? Remember, it's the IgA that travels in our bodily secretions like breast milk. Okay. Breastfed. Okay. An artificial active immunity, that's when you get some kind of vaccination. All right. So we are artificially exposed. We get some kind of jab, usually a shot. And that's how we teach our B cells and T cells to make memory cells, so that the next time we get exposed to that particular pathogen, we kind of have a head start on the immune response there. So we already have some, very quickly start making some antibodies and, hopefully activate T cells as well. So we don't even know that we've been exposed. And then lastly, an artificial passive immunity. Sometimes we will give patients infusions of antibodies. We've been doing this a lot with COVID, people that are particularly at risk that maybe contract COVID. We can kind of catch it early enough or get them treatment early enough. If we give them some antibodies to COVID early on, then we think that has some help in them recovering more quickly and less likely to have some bad complications. So, okay, we've done that with other infections like Ebola. Didn't do a lot of help for Ebola patients, but basically those are the ways that we get antibodies. I had about a year ago, I had a friend tell me, well, the whole, vaccination thing, which we'll get into here pretty soon. But she said, well, I don't want to be vaccinated. I think eventually we'll all just have antibodies to COVID. And I wanted to say, okay, these are the 4 ways you get antibodies to something. Okay, you either get the illness, you get the vaccine for the illness, you get antibodies artificially, or you find someone that's, lactating and is willing to let you. Okay, so you're not just going to breathe in and, get antibodies to this. So they're very, specific ways. So, we hope that eventually there's really mild forms of COVID and you may get sick and, not even know that you were sick and, have some antibodies that would help you fight it. If it, did mutate to get a little bit, but we don't know. We don't know what's going to happen with that. So, okay, so here's a practice question using those terms. Once you get them down, you'll have them. Okay, so take a minute, read that over, make your choice. So we are, in this example, exposing a horse to botulism in some kind of small amount that won't kill it, obviously, letting the horse make antibodies to that, and then harvesting those antibodies from a horse, giving them to a human. So that's going to be not natural, right? That's going to be artificial. And then when you just give somebody antibodies, right, that's passive because the person's not making the antibodies themselves, right? So artificially acquired, passive immunity. I think there's a few homework questions that let you practice with that as well. Let me give you one other practice question. I've asked this question on exams before. I love this question. But it is a little bit more on the difficult side. So I'll let you wrestle with it a minute. Again, this is just reviewing from last time. Thoughts? So I like, you didn't really have to know anything about malaria, but I like this question because we did talk about malaria. But what's special about red blood cells? Right, they don't have a nucleus, right? So cells that don't have a nucleus don't display antigen by MHC class 1. Okay, all right, so correct answer there is A. So they don't really have any way to advertise to our cytotoxic T cells that there's a problem in there. So that's one of the reasons that we have hard time, immune system has hard time with malaria, and it can be quite severe. Okay, so it's very easy for me to make questions like this, especially with adaptive immunity, because it's easy to think of a lot of things that kind of sound good, but that aren't the right answer, right? So, you know, we have lots of different cells that we talk about their function, and, you know, just try to keep straight what kind of cells do what kind of thing. If I say something like, oh, the T cells can't make antibodies, or nope, not T cells that make antibodies, remember, T cells. So kind of make a lot of things sound good, but yeah, just think about it a little bit. Okay, questions? Yeah, okay.
So if it's like we can't like that kind of thing necessarily to the malaria, like what medication do they use? I know they use a medication to like prevent it from happening, like when you travel to Africa, like how does that medication work?
All right, so great question. So you said that, okay, if an adaptive immune response is prevented, right? So how does the medication that keeps us from getting malaria work? Does anybody know? That's a great question. I have not really investigated the drugs that we have available that are supposed to be. So that's a great question. We are going to talk about a vaccine today and how that works, but I would have to look into that, how those anti-malarial drugs work. So good question. Yeah.
Yeah, because I know a lot of times with like autoimmune diseases, they prescribe anti-malaria.
Interesting. So for some autoimmune diseases, they prescribe anti-malaria.
Like I know for lupus.
Okay. Oh, okay, interesting. Okay, good. Okay. All right, so moving on. So this is a picture that I did not post. This is a little introduction here to talking about vaccines. I've always, this just came from the Anti-Vaccination Society, something that I just Googled and came up with. 1802 was when this little cartoon was published. And I think it's so interesting. Of course, it refers to Edward Jenner's smallpox vaccine. The first vaccine that we developed, it was a process he called variolation. So basically, what Edward Jenner is credited with figuring out is that milkmaids that had been infected with cowpox, which, by the way, I've seen cowpox lesions and they're pretty nasty. It's not like just some little rash that you get. But he noticed that milkmaids that had cowpox never seemed to get smallpox. And so he reasoned that maybe cowpox was kind of a milder form of smallpox. And what he did to test that theory is that he scraped off some lesions from somebody that had cowpox and then inoculated that scab into a young boy's arm. So they called it variolation. And then a few weeks later, he intentionally did this, took somebody that had smallpox scabs and then jabbed them into the little boy's arm. So, not really how we do clinical trials today, right? We'll get to that. But the boy didn't get sick. And so it became a common practice to try to, keep yourself or your loved ones from getting smallpox. And there has always been some resistance to vaccination because especially back then, people didn't understand why that worked, why it kept people from getting smallpox. So you see in this picture, people getting inoculated, and then there's other people that have, like here, this woman's throwing up a cow. It looks like she's giving birth to a cow down here. This guy's got a cow growing out of his face. This guy's got problems down here. So it's natural to be afraid of things that you don't understand. I don't think they thought that those things would really happen, but the point was, hey, we don't know what effect this is going to have on us. Vaccines are a lot safer than they were back then. I mean, back then, yeah, it was, smallpox had about a 30% mortality rate. So that was pretty high. So you were still better off getting vaccinated. But it did have its risks, especially with, getting some kind of introducing some pathogen, just by, using a knife basically to inoculate yourself. So we've come a long way since then. We'll talk a little bit, just a few terms here, why we vaccinate. We'll talk about the different kinds of vaccines that are in use today. So that includes the COVID ones and this new malaria one. I haven't updated anything really since last fall. So right when I was talking about this last fall is when Aaron Rodgers came out and, he had just gotten COVID and how he had said, well, I was immunized. Well, what did he mean when he said I was immunized? Isn't that the same thing as being vaccinated? Most of us would say, yeah, those two things are the same thing. Vaccination is basically what happens in your body. Basically, you gain immunity when you are immunized, meaning you get a vaccine. So yes, we get vaccinated by immunization. What Aaron Rodgers was calling immunized was, I took some kind of herbal remedy and it boosted my immune response. Okay, that's not the same thing as getting a vaccine. A vaccine is, boosting your immunity to a specific pathogen, getting memory B cells and memory T cells. Okay, so again, usually we use those synonymously. Why should we vaccinate? So I'm not just talking about COVID, we'll talk about that more. But in general, our goal in vaccination is primarily been to protect young children. We talked about how there are some pathogens that are the immune systems of young children don't recognize very well. Those B those T cell independent antigens. Young children don't have a very good response to that. They just have an underdeveloped immune response in general. So especially babies are a lot more susceptible to a lot of different infections. So we want to protect them from those childhood illnesses. Remember in 1900, we said about a third of kids died before their 5th birthday. Okay, that was before we had, I think we had a couple of vaccinations then, but they were not widespread. Okay, so we want to protect children. As we age, we know that our immunity wanes. So there are a lot of vaccines that people say over the age of 65 are recommended to get because they're more susceptible than people that are younger. You've heard a lot in recent months, maybe about herd immunity. And will we get to that with COVID? What that refers to is that if a certain number of people in the population either become vaccinated or become immune to an infection, then the people that are not vaccinated are protected. Okay, so in other words, if, let me write that, if most of the population is vaccinated, people that are not vaccinated are protected. Because not everybody can be vaccinated. There are people that either they're babies and they're too young and it won't do any good, or there are people with immune conditions where autoimmune or other immune diseases where it doesn't do any good. Maybe their body can't generate those memory cells. There are people that are allergic to something in the vaccine and they just can't be vaccinated. So the idea is if most of us, and you know, what percentage has to be reached to reach that herd immunity, it depends on what infection that we're talking about. But if most of the rest of us get vaccinated, then the people that are not vaccinated are just not likely to come in contact with the pathogen. So they'll be protected. Recently though, we haven't heard a lot about herd immunity. We were hoping that we could get to that with COVID, and I think that's not going to happen. Like, not enough people are willing to be vaccinated, and plus we would have to vaccinate the entire world, right? So it's, we haven't even been able to get high enough vaccination rates in the US, let alone lots of other countries. So you hear less and less talk about that. We think that, like for influenza, that herd immunity may only need to be like 30 or 40%, so it wouldn't be spreading. Other things like measles, it has to be like 95%. If we get less than a 95% vaccination rate, it starts to spread in the community because it's so infectious. Okay, so the goal of vaccination, we want to induce an adaptive immune response. Okay, so we want to make memory B cells and T cells. Okay, so ideally we're going to get both a cell-mediated response, cytotoxic T cells, as well as a humoral response, lots of antibody production. It's usually a little bit easier to get that humoral response because it's sometimes difficult to get our cells to take up a pathogen that's not alive. We don't get quite as good a T cell response, but usually at least we get a humoral response that's oftentimes good enough. All right, so let's talk about the different kinds of vaccine. This used to be a much shorter discussion than what it is now. There's basically 3 kinds of vaccines that we've used for a long time and that have been approved by the FDA. The very first vaccines were attenuated vaccines. And that means that they contained a live pathogen, but attenuated means weakened. So some kind of weakened strain so that it couldn't cause full-blown disease. So that agent, whether it's a virus or a bacteria, has been modified a little bit so it's not as infectious. How do we do that? Sometimes, like for example, and again, I like to give you a lot of examples of different vaccines. Don't worry about memorizing, the example, just kind of know the principle, what an attenuated vaccine is. We have a couple different kinds of polio vaccines. One of them is an attenuated vaccine. It's called the Sabin polio vaccine. And it's the polio virus, but it's been weakened because we've grown it in monkey kidney cells, which is not its normal host cell, instead of human nerve cells. Okay, so that weakens it, causes mutations to build up, so it's not as virulent. Sometimes we grow it at a different temperature. Maybe sometimes just growing it for many generations has allowed mutations to kind of build up, and that it's just not causing as a full infection. So there are some advantages with this kind of vaccine. It's going to kind of grow and multiply naturally in a host, so we get, just exposure to lots of different epitopes of antigens, and we get a more natural adaptive response. Our immunity is more likely going to last longer, and it takes fewer doses. So you may only need a couple doses of it in order to get lifelong immunity. And this can be considered an advantage, can be considered a disadvantage too. We'll talk about that. But some attenuated vaccines have actually caused a mild form of the disease. So then say you have a population that, it's maybe very rural, and you can't get to everyone. Maybe it's, a village high up in the mountains. If you vaccinate part of the population, maybe those kids will play with the kids that didn't get vaccinated and spread some contact immunity. So they could spread a mild form of the disease even to people that weren't vaccinated, and thereby, getting more people protected. Okay. All right. So, that could be a positive. If you think about the negatives to attenuated vaccines, that's really disadvantage too, that it's possible to have transmission. Because, again, although you could think of that as a positive, we don't really want to be doing that in this country, because, you don't want to pass it on to somebody that's, maybe immunocompromised, or to a newborn baby, or to somebody that may, whatever, have some reaction that you didn't anticipate. So generally, we don't want that to happen. It's a little bit more expensive to transport these kinds of vaccines because they have to be refrigerated. Again, because there's some, live agents, so you have to keep them refrigerated. So that's going to increase your costs. And really, the biggest disadvantage is that the very slim possibility of reversion back to a wild type. Most infectious form. So we didn't really worry about that too much for a long time, until it happens once. So like I said, there's two different, for example, here, there's two different vaccines for polio. And they both came out about the same time in the 50s. One was a killed polio virus, so we'll talk about that in a second. The other was this attenuated strain. We kind of went back and forth about which one that we used. Finally decided to use the Sabin one, the attenuated form, worked a little bit better, less doses needed. And in fact, babies could take it orally. So it came in this little plastic tube, and you could just get babies to drink it. And one less needle prick sounds like a good thing. Well, until the year 2000, they administered a lot of this vaccine, a certain batch down in Haiti and the Dominican Republic. And they had a number of children that came down with polio from the vaccine. Tragic situation. I don't know if any of them died or not, but somehow they got a batch that had reverted back to that, most infectious polio. So for that reason, we don't use that vaccine anymore. We use the other one that maybe is not quite as good, but again, there's no possibility that you can get polio from it because the agent is actually killed. So in general, we don't use attenuated vaccines unless we don't have anything else that works. Okay, we've tried a measles version that was inactivated virus, and it just didn't provide good enough protection. So like the MMR vaccine, those are all attenuated. You can get a flu vaccine that's attenuated, but that's not the most common way to get a flu vaccine. I believe chickenpox is attenuated vaccine, so there are some out there. More commonly, though, we have killed vaccines. We also call them inactivated because, well, is a virus alive to begin with? Usually not. We would say usually not. Okay, and there's two kinds of killed vaccines, either whole cell or subunit. Okay, so whether you're talking about whole cell or subunit, again, some good things, some bad things about this kind of vaccine. They don't actually replicate in the host, right? Because they've been killed somehow. And we typically have to give more boosters to get the same level of protection. Mostly, they're going to give us a humoral response. Because they're not alive, our cells, say if it's a virus, our cell is not going to take it in like it would a live or active virus, and it's not going to present that antigen with NAC class 1 in the same way. So we don't get as good a T cell activation. Usually it's a little bit easier to get our B cells activated to make antibodies. Usually that's good enough. And there's lots of examples of this. The Salk polio is the one that we use now, rabies, most of the flu vaccines. Pertussis, we'll talk about that in a second, otherwise known as whooping cough. All right, so if we use the entire pathogen, the whole agent, okay, we have to kill that somehow. Usually that's done either by maybe hitting it with gamma rays. Remember formalin, that was an aldehyde, so a pretty high level disinfectant. It only takes a tiny amount, and it's enough that's safe to be included in the vaccine. Sometimes they can heat it. I would think often that would denature proteins that would be vital, but sometimes, whatever works for that particular pathogen. The big drawback with a whole cell vaccine comes in the case where you have a gram-negative pathogen. Okay, so remember what's on the outside of the gram-negative cell wall. That lipid A, we also call that endotoxin, right? If it's the whole cell, the endotoxin, even though it's dead, the endotoxin will still be somewhat intact. Okay, so with the case of pertussis, which is a gram-negative pathogen, when we gave that whole cell, we would give that pertussis vaccine years ago. It was called DTP. All right, so the D stands for diphtheria, the T for tetanus. We give that in conjunction with the pertussis whole cell. Well, a number of years ago, I think it was about 20 years ago, we decided to change and not use that whole cell pertussis anymore because it caused a good deal of inflammation, especially around the site of injection. Okay, now this is not a serious side effect. things like, redness, swelling, maybe a little fever as a result of that. But when, that pertussis is especially important to give to babies. And if a mother comes in, the baby gets their pertussis vaccination at 2 months, and then the rest of the day, the baby's, screaming and upset, is the mom going to come back and get the next one? Okay, sometimes not. So that was getting to be a problem, that little extra inflammation from the endotoxin. So about 20 years ago, they switched to using a subunit vaccine. In other words, sometimes we call them acellular. So a subunit vaccine just has a piece of the pathogen. So maybe oftentimes it's something from the surface of the pathogen, important protein, maybe that's antigenic. So there's no way that, that can cause disease, but it's enough to kind of train our immune system. So problem here, even more boosters are necessary because there's just not as many epitopes for our immune system to learn how to recognize. So we often have to give more boosters, but good thing is less problem with inflammation. So now we give that diphtheria, tetanus, and then the AP, that stands for acellular pertussis. I think we've been doing that for at least 20 years. And it's that, we do have some problems with kids getting pertussis when they're, even 12, 13 years old, even when they've been vaccinated as babies, that immunity wears off. But getting pertussis as a, adolescent is not as serious as getting it as a baby. So, third kind of vaccine that's been in common use for a long time are the toxoid vaccines. So some of our infections, it's really the toxin, the exotoxin produced that causes the disease. So it makes sense to have our immune system learn how to neutralize that toxin, to make antibodies that will just coat that toxin to neutralize it. So they inactivate the toxin. Again, they add some formalin or they heat it or hit it with some radiation. We get a really good antibody response. And again, that's usually good enough. Tetanus and diphtheria are the two toxoid vaccines. So again, a toxoid is just an inactivated toxin. That pertussis vaccine, that acellular pertussis, actually includes some pertussis toxoid as well. So, right. And this is just a picture of somebody with diphtheria. That's not an infection that we hear of anymore because we have a good vaccine to it. That Corynebacterium diphtheriae makes this like pseudomembrane in a patient's throat. You can see this kind of whitish mass back here. It actually will choke out somebody so they can't breathe. And you would think, oh, can you just scrape it off? But you can't. So again, a childhood illness that we don't have to think about anymore because we do have good vaccines. All right, so in terms of what vaccines have been approved by the FDA, our list is a little bit longer now. There has been work with using messenger RNA technology for 30 years. So, people call this new. It's really a technology that's been around for a while. It has a lot of possibilities in terms of using messenger RNA as drugs, in terms of using it to treat cancer. But here we'll talk about using it as vaccines. And this company, Moderna, it was formed, that name comes from modified RNA. Okay. So some advantages to this type of vaccine. Because the vaccine, it's just basically a piece of RNA that's been kind of doctored with a little bit. It's very safe in terms of the patient. There's no chance that the patient can get the infection that we're trying to vaccinate against. It's not going to mutate the patient in any way. We don't make DNA from RNA, remember, ever in our cells. And it's really easy to make them. Within a few days of after we knew the genome of COVID-19, a lab had already made a RNA vaccine. Like that's how flexible and easy it is. Then we had to test it for like 8 or 9 months to make sure it was safe. But we can rapidly kind of change that RNA a little bit. We can do that really quickly. The way we make flu vaccine now, it takes about 10 months to make it. So we have to decide in February what strains to include, and then we have to grow it up in eggs, and it takes a lot of time to get ready before the next flu season. So they're easier to make. One disadvantage in terms of trying to make the vaccine and figure out how to do it is that messenger RNA degrades really rapidly. It only stays in our cells maybe a day or two. So there was a lot of work that had to be done to figure out how to get it into cells, so our cells would let it in, and how to keep it from degrading quite so quickly once it was in there. So one of the reasons, though, we have to keep it very cold to keep it from the vaccine very cold to keep it from degrading. And another problem is that very short-term inflammatory reactions are common, right? Most of us know that. Probably most of us got a little bit sick when we had that vaccine. If you think about it, we really don't want a big innate reaction, right? You have to have some innate reaction in order to get your adaptive immunity, but we don't want all the inflammation it's a common part of a normal immune response. So those nucleosides that were put into the messenger RNA, they've modified them a little bit to try to make it so our toll-like receptors 7 and 8 don't recognize it. So if you remember, those toll-like receptors, they're on the outside of phagocytic cells, and they recognize PAMPs, pathogen-associated molecular patterns. All right, so the single-stranded RNA acts as a PAMP. So when the phagocytic cell phagocytizes it, this TLR8 and 7 and 8 are actually expressed in the endosome and recognize, the insides of pathogens. And once they are recognized that, then they kind of they basically activate lots of aspects of our innate immunity. So one of those things are inflammation. So if we can try to kind of hide that messenger RNA from our toll-like receptors, we found that's been helpful. So a little short-term inflammation for a lot of different people. A more serious effect has been very rare is that myocarditis. So that's inflammation of the heart. Usually manifests as a little bit of chest pain, usually goes away after a couple days. But they found after the second shot, about 1 in 50,000 people can develop that. There was just an article that came out this week from the CDC. They've done an exhaustive study of that and found that myocarditis in people that get COVID, it's about twice as common. Okay, so yes, it's possible to get it from the shot, and unfortunately, it's mostly young people that would get it from the shot. But if you get COVID, you're twice as likely to get it. So you're always weighing the risks. And again, this is not a super adverse effects most of the time. The other vaccine that maybe some of you got to COVID was the Johnson & Johnson vaccine. And of course, this is a slightly different vaccine. It's called a viral vector vaccine. So what we've done in order to trick ourselves into taking up this piece of, a little piece of DNA that encodes for the spike protein, right? I forgot to say that. For the RNA, we use a piece of RNA that encodes for the most antigenic part of COVID-19, the spike protein. So as a way to get it into the cell, they've used actually a virus. So we've used, in this case, we've used an adenovirus as a carrier. Blanking out, I think there's a couple different, the AstraZeneca, the Oxford vaccine also used an adenovirus. One of them used adenovirus from a chimpanzee, so it was one that wouldn't ever cause disease in us. I can't remember if this was a human adenovirus that they used, but it's a virus that doesn't replicate in our bodies, or it's been altered so it can't replicate. And even if it did, it would just cause like a cold maybe. So that's how they got that piece of DNA into the cell. And then basically, so like in the RNA, it teaches our cell to make that, that spike protein. So some of our cells will actually manufacture the spike proteins on the surface. And in addition, hopefully present some spike protein by the MHC class 1. Sorry, not 2, MHC class 1. Okay, so that's the whole idea, to get our cells to not only express some spike protein, but present some of it by MHC class 1. And in this way, we get different aspects of our immunity involved, our immune cells, our adaptive immune response. Okay, so we didn't have to refrigerate this one quite as cold. DNA is a little bit more stable than RNA. A single dose, you know, for a while we said that was good enough. Now I don't know what we're saying exactly about that. The Johnson & Johnson shot has really fallen out of favor because it has some more potentially serious side effects. This thrombosis with thrombocytopenia syndrome is a rare clotting disorder. Incidence of that, they've said 3 in a million. So it's rare enough that, you know, there's some question as to, is it really the vaccine that's causing that? But certainly they're trying to be, you know, upfront about that. This Guillain-Barré is a little bit more common, about 1 in 50,000, I think. Let me double check that. Oh, no, 1 in 120,000. And Guillain-Barré can be a potential kind of side effect of any viral infection. So it's not just vaccines that can cause this, it's kind of a neurological disorder that can cause some temporary paralysis, but usually clears up. Okay, so a little bit more serious, again, potentially serious side effects, still very rare, still much more dangerous to get COVID in general. So when those vaccines came out about a year ago, a little bit earlier than that for some people, of course, people worried that they were rushing through. So I think it's important to think about, well, how are vaccines tested in this country? So this is just a screenshot from the CDC's page about how vaccines, what kind of testing that vaccines go through. So they actually go through 4 phases. So we'll talk about these 4 phases. Before they enter clinical testing, of course, they have to be tested. I guess they don't talk about that there. But before they even enter this, they're going to be tested rigorously in animals. And sometimes we don't have a good animal model for a vaccine in terms of causing that same infection in humans, causing that same infection in animals. But at least we can test the vaccine in animals and make sure that it doesn't make them sick in any way, have some side effect. So lots of testing in animals. And then when they're, we think that all that data looks good, they start the human trials. So in phase 1, you start with volunteers, small groups of volunteers. So maybe somewhere between 20 and 100 volunteers that are healthy. Okay, so they're checked out to make sure that they don't have any pre-existing conditions. They look for side effects. Sometimes they can tell, hey, is this effective? Do the person, they're not intentionally exposing those people to the pathogen. but they see if, you know, they contract it when they go about their business. Okay, they look at that data. If that looks like there's not any, you know, side effects that are coming up, they'll expand that into several hundred volunteers. And these are not just people that are considered the picture of health. They're people that might have, you know, certain, they might be diabetic, or they might have, you know, whatever, other things going on. They'll mess with the dose a little bit. And just, again, trying to get an idea, is it effective? Are there side effects? Okay, if that data looks good, then they'll expand it into thousands. I think Pfizer had like 30,000 at least in their phase 3 trial. And of course, some people are going to get a placebo. Okay, they got to have controls on there so they can understand if it's effective. All right, is it actually keeping people, you know, how effective is it from preventing COVID or whatever disease we're talking about? Okay, so at that point, they have to wait at least 2 months after they get after they infect those thousands of people, because side effects from a vaccine are usually going to show up in 2 weeks. So they've given, you know, extra time, 2 months. And at that time, if everything looks good, then the FDA will approve it and it will be started using in the general population. Okay, so none of those steps were skipped with these vaccines. I understand that they did kind of start phase 2 before phase 1 was quite done, and they started phase 3 right before, you know, they kind of squished them together, but they did them all. In Russia, they started administering to the general population after phase 2. They didn't even do a phase 3. So that's why a lot of people were not willing to get the vaccine in Russia. Okay, but there's actually then a phase 4, which is ongoing. Okay, any vaccine that we're using in use today is in phase 4, and that's the VARS system. Oops, what happened? Won't do that. There we go. The VARS system. Oh, hopefully my daughter's not calling me. So that VARS stands for Vaccine Adverse Effects Reporting System. Okay, so this is in the past, basically, if your baby, say, got a rash after a vaccine, you would call your doctor, or you would call the number that you got on the information sheet after the baby got the vaccine. Today, we can be even more flexible. I signed up for this when I got my COVID vaccine, and then I don't know if some of you did that too. Like every day for a week or two, I would get a text, and they would say, "How are you feeling today?" And you know, I was like, "Oh, I'm fine, thank you," or whatever. Report any symptoms that you have, just to be a data point. I just got a text last week. It had been the 1-year anniversary of my second shots, and they wanted to know how I was feeling and if I was in generally good health since my last vaccine and all that. So we have a rigorous reporting system in place so that we can catch what side effects there are, and we can just kind of understand that better. Okay, so that's ongoing. Questions, comments? Okay, there's one other kind of vaccine that I want to talk about. You know, I often will have the discussion, well, why don't we have vaccines for everything? Well, we're trying, but there are some infections like HIV that they're just constantly changing so quickly that we haven't been able to develop a vaccine. Gonorrhea, we're going to talk about that pathogen next week. It just changed. It mutates so quickly that we can't even get memory cells that would do any good. And it used to say, malaria is the same way. There's just so many different strains of that Plasmodium pathogen that a vaccine is just not feasible. Well, it turns out that the World Health Organization, the WHO, approved a vaccine last fall for malaria. So I didn't even realize that one was in the works. But this is it's a recombinant vaccine. So I included it on this list. Again, we're not using it in this country. It's not approved by our FDA, but it is in use and will likely be in more use. A recombinant vaccine, what does that mean? Well, they've taken parts of pathogens. So in this case, it's part of this. Okay, let me write malaria on here because I actually never wrote that anywhere. They've actually taken part. Remember the pathogen that causes malaria, Plasmodium falciparum. There's this protein. Remember, malaria has all these little stages that we've got all this terminology for, but this is a stage of the malarium right like before it enters our liver. Remember, that's the first stage. After it matures in our liver a while, then it enters the red blood cells, and that's where it starts to do all the damage and causes the high fevers and things. So they took a little bit of this protein, or in other words, they isolated the gene that makes that protein, and then they grew up, they recombined that with the DNA from a yeast cell. It's either yeast or bacteria, I'm not sure which, and taught that yeast or bacteria to express that gene, and then we harvested that protein. Okay, so that's kind of what happens with a recombinant vaccine. We kind of grow up parts of the pathogen in a yeast or bacteria cell. So the name of this vaccine is not intuitive at all, RTSS. Well, the R and the T refer to this protein that's, again, on the surface of malaria. Now, the problem with just using that as, say, like a subunit protein is that it's not very antigenic. Our immune system doesn't recognize it very well. So what they had to do to boost the immunity is to use a surface antigen from hepatitis B virus. Now, I don't know how they chose hepatitis B virus, but they picked a protein from another pathogen that does cause a good immune response, and they express that as well. So that's what the S is from this hepatitis B virus. So anyway, they threw all that together to try to prevent, get our immune system to prevent those Plasmodium protozoa from entering our hepatocytes, our liver cells, to begin with. Okay, and it works to some extent. So it induces enough antibody to keep that from happening part of the time. How effective is it? About, in their trials, about 30% effective in preventing severe malaria. Okay, and mostly they're giving this to kids because kids are the ones that are more likely to die. Okay, so if a kid gets severe malaria, they're likely to die. So normally a vaccine really wouldn't be used or accepted if it was only 30% effective. But in this case, 30% effective means it's saving 3 out of 10 lives. So it's been used, kind of on a trial basis for several years now. And unfortunately, it does take 4 injections. So difficult maybe to get, kids a series of 4 injections in sub-Saharan Africa. But there are, sort of better variations of this in the pike, and they think that we're going to get better and better at this. And so hopefully we can improve that effectiveness. And again, that's going to be a good thing. Okay. So just to kind of wrap up talking about vaccines, we talked about kind of the different kinds that are in use. It's really no arguing with the potential, the tremendous benefits that vaccines have been for us. Really, they're considered the most efficient, we'll say cost efficient way to prevent infectious disease. You really can't argue with that when you look at data of certain infectious disease. So in this case, polio. Polio was never like extremely common. At its height, maybe we had like 40,000 cases in the US. But with the introduction of two different vaccines, we haven't had a case of polio in the US since the 1970s. And in fact, it's been eradicated in every country of the world except Pakistan, Afghanistan, and Nigeria. So we're really close. There's only about 200 cases a year. Probably COVID has put a little wrench in trying to totally eradicate it, but we're really close to that. So that's exciting. Of course, smallpox was eradicated. Measles, you know, that used to be something that everybody got. So came out with a good vaccine in the 1960s. You know, people, you know, your grandparents, they probably all had measles. And there's complications aren't common in this country, but worldwide that still causes a lot of deaths. So, you know, it's hard to argue with, you know, having a measles vaccination. So, you know, there will always be the argument about personal freedoms versus, you know, your commitment to not pass it on to somebody else that's vulnerable. Anyway, I think, you know, on the side, I'm kind of pro-vaccine, I guess you could say. And, you know, hopefully people just take the time to understand. I was talking about side effects. I was going to one prevalent myth that you hear is that the COVID vaccinations can cause infertility, right? So this is something that doesn't have any basis in any we don't have any evidence from that at all. And I don't call myself an expert in immunology, but Dr. Renkema, who is an immunologist, she's looked at the data for that, and she said it's just ridiculous. Like there's no mechanism where that could happen. So if that's something that you're worried about, definitely, you know, look into it, look at the data, you know, inform yourself. So questions, comments? Oh, yeah. So that's a good question. So because their Pfizer and Moderna are mRNA vaccines and they degrade fast, is that why we need boosters? I don't think that's the reason. I think it's just, yeah, I think it's just a matter of not getting enough. You know, we just have, it's just one protein on the surface of COVID, and I just think it's not antigenically very diverse. And so yeah, I mean, that's a great question. I don't have a great answer to that. You know, we were hoping we'd have, you know, 1 dose or 2 doses, and then we wouldn't have to have any more. Of course, this week we just, you know, it was just FDA approved a second booster shot for people that are older than 50. You know, that's a good question. Is this going to keep going on and on? It may be looking like we may need our yearly COVID shot. Yeah, it's a great question. I don't think it's just because it's messenger RNA though. What's that? Well, right. So that's a huge problem. RNA viruses often mutate pretty quickly. And when you have that many people infected, then you're going to have some mutations that are going to make it more successful. So yeah, definitely. Yeah.
So the difference between the mRNA and the viral vector is they have the same mechanism. It's just based on like what kind of material is put in the cell. Right. So the difference between them is how they get them to enter into the cell. Which is just like what bacteria pages with mRNA.
So I think I didn't talk about that, but the Moderna and Pfizer use nanoparticles. They're just like little lipids that help ease it into the cell. That's my understanding. Instead of using a virus.
Just both cause them to express the virus.
Yes, they both cause. Yeah, they both. That's the same. They both cause, you know, some of our cells to express that spike protein from coronavirus. So good. All right, so let's start our staff. I knew I would end up taking a lot of time on not very many slides there for our vaccine stuff. So now we're kind of starting into that ending of the semester where we're going to talk about some specific pathogens. So a lot of people kind of like this talking about diseases kind of approach to things. It also is kind of a nice opportunity for us to kind of, hey, remember we talked about this, you know, this organism does that. And so it's a little kind of nice way to wrap things up. So we're going to start with the gram-positive bacteria, the medically important ones. So we're just going to kind of pick and choose different ones that are in your book. So I've just referred to, you know, where you can read about them in your book, but concentrate about what we talk about in class. The cocci that we think of as being important, medically important, are the staph, the strep, and maybe to a lesser extent, the enterococcus, which of course the enterococcus are very similar in a lot of ways to streptococcus. So it's nice that we're back to kind of crossing over with lab a little bit. So if you're in lab, a lot more of this will look familiar, but I try not to ask you things in lecture that I think, oh, they know that from lab. I try not to do that because I know some of you are not in lab. So, all right, so let's talk about this genus Staphylococcus a little bit in general. In lab, of course, we focus on 3 different Staphylococcus. There are more than 40 different species of staph out there. There's really one that's really most famous in terms of, you know, clinical implications. So we're going to focus on that one, Staphylococcus aureus. But in general, what would we say about the staph? They tend to arrange themselves in grape-like clusters. So remember, I'll come back to this. Whoops, I'll come back to this picture in a minute. But remember at the beginning of semester when we were, you know, just describing kind of different attributes of cells and how different terminology for how they arrange themselves. Our Staphylococcus make these grape-like clusters. As we'll see, the Streptococcus tend to divide only in one plane and then stay stuck together into chains. You may notice that if you did a gram stain in lab this morning, these nice chains, that probably was a Streptococcus if you saw that. And then there's also, you know, tetrads possibility or kind of cube-shaped arrangement that we refer to as Sarcina. Okay, so that helps us kind of, again, characterize certain organisms or certain genus. Where do we find them? The staph in general are common on our skin, in our upper respiratory tract, like our nose. And they're going to be common places that we touch as well. So especially indoors, any surface that you touch is probably going to have staph on it. What characteristics do we commonly see? They make an enzyme called catalase. And we've talked about in here the importance of catalase in terms of a detoxifying enzyme so that peroxide ion that can be produced as a normal part of aerobic respiration. If organisms can produce catalase, they can detoxify that. And what you end up with is water and oxygen. So let me balance that. All right, so of course in lab, when you're trying to see if your organism makes catalase or not, you rub a little bit on a slide dry, and then you add a drop of hydrogen peroxide, and if it bubbles, if you see this oxygen come off, then you've got an organism that makes catalase. And this is a really important test, of course, because it helps us distinguish from the staph and the strep. Okay, the streptococcus don't make catalase. There's lots of organisms that make catalase though, so don't say, oh, if it makes catalase, it must be staph. Remember, lots of gram-negatives make catalase. Bacillus makes catalase, Clostridium doesn't. So it's a very common enzyme, but it is, again, helpful to distinguish between strep. Some of our staph will form capsules. Often it's maybe kind of a wimpy capsule, you know, or we'll say, or a slime layer. Okay, so tends to be some of the straps form a little bit beefier capsules. Most of them are considered facultative anaerobes. So that means they're very flexible, they respire aerobically, they may have some kind of anaerobic respiration or fermentation that they do as well. And we'll say they are a little bit more environmentally hardy. So I'll put that in quotes. What do I mean by hardy? I don't mean they do not form endospores. They do not have an acid-fast cell wall. They're not that hardy. But we do see that they are pretty good at surviving fluctuations in pH. They survive at lots of different temperatures. They're pretty salt tolerant. So they just survive a little bit better, certainly, than the Streptococcus, which is often what we kind of compare them to. The straps are pretty wimpy in the environment. So by the environment, I just mean outside of our body. So on surfaces or, you know, potentially outdoors as well. All right, so like I said, let's zero in on Staphylococcus aureus. Aureus is Greek for gold. So sometimes the colonies are a little yellowish. We try to use strains that are not very yellow because we don't want to make it too easy for you to identify them. But sometimes you see that. And in terms of the epidemiology, in terms of where do we find them? What's the occurrence? How do they spread? Staph aureus can be considered, for some of us, part of our normal microbiota. So about 30% of us, we could, you know, test and find that we're carriers, at least transiently, for Staph aureus. Sometimes we may consider them an opportunistic pathogen. And scientists argue about that, whether to call them a true pathogen or an opportunistic pathogen. But it takes a pretty big infectious dose through whatever portal of entry, you know, whether you swallow it or in a wound, for instance, to actually get us sick. So that's why sometimes it's considered a true pathogen. Some people say not, because it does take a big infectious dose. We do know that commonly especially the antibiotic resistant strains are commonly transmitted in hospitals, but really any close contact in the community. So I'm talking about, you know, jails or health clubs, or, you know, anywhere where people are in close contact with each other, you know, locker rooms, sports teams, then Staph aureus infections can be spread. A lot of staph infections are skin, but there's some other more invasive infections that are possible as well. So we'll talk about that. All right, so thinking about what kind of tools that Staph aureus has that make it successful in establishing an infection. So we call those virulence factors, right? And not every strain of Staph aureus would have all of these. So some strains, you know, might have some of them and some have the other. But it is pretty common for them to have some kind of capsule or a slime layer. Remember the benefits of that. It keeps it from being ingested or phagocytized very easily. And it really is going to help it adhere to some kind of structure. So some kind of implant or, you know, during surgery or a catheter or whatever, it's going to make it more successful. And another structural virulence factor, this will be new, we call it protein A. Okay, so some strains of Staph aureus will have protein A, and if you can picture it, like it will have protein A, you know, covering the whole outside of the cell. All right, so picture that in 3D. We'll have protein A all over the outside. What good does that do? That actually binds to the FC region of an antibody. So remember, if this is an antibody, up here at the ends, that's the fragment of antibody binding. And then the stem region, that's the FC portion. Okay, so it's up here at the fragment of antibody binding where the antibody is going to work to, bind and neutralize to adhesive proteins, adhesion proteins, for example, or bind to a toxin to neutralize it. If right here, that antibody is bound kind of by its stem region to that cell, then that fragment of antibody binding is basically, it can't do any good because that's the end that wants to bind to, if it's bound this way, okay, now a pseudopod might have a receptor for that and would be able to get a hold of that cell much more easily. So it's kind of like grabbing a snake by the back of the head, right? Then you're not going to get bit. You can grab it by the back of the head. So if you're grabbing it by that stem region, then do any harm. All right, we're out of time, so we'll continue. We'll finish up staff next time. That will be the end of your Monday's exam material, and we'll start strep deposit.